For our post-lab discussion, we will be talking about the organisms associated with your water analysis and what other tests we can use to determine them. For our post-lab discussion, we will be talking about this. So drinking water is a clear, colorless, without disagreeable taste or odor. It has your saprophytic bacteria, which are your Pseudomonas, Serratia, Flavobacterium, Chromobacterium, Acetanobacter, and your alkali genes. Saprophytic meaning they do not cause harm. In the community, you have your bacteria, your Shigella, your Vibrio cholerae, your Salmonella, and your Leptospira. For the parasites, you have your Georgia lamblia, Amoeba histolytica, these are your protozoans, and your helminths, which are your schistosomes. And lastly, for your virus, we have your enterovirus. In the hospital, we have your Legionella pneumophilia, your Serratia marcescens, your Mycobacterium schinopi, Pseudomonas paucimobilis, Mycobacterium chelonae, Acetanobacter, and your Pseudomonas aeruginosa. For the bacteriological examination of water, again, this is a test for fecal pollution. So it detects the presence of your coliform bacteria. Coliform, coli from your colon. So mainly feces yung bacteria niya. So it's easily detected even in high dilutions. So your bacteriological examination of water as a confirmation test to detect of the thermotolerant Escherichia coli. So this is a definitive proof for fecal contamination. Other bacteria than E. coli can be seen as your fecal streptococci and your Clostridium perfringens. Bacteria growing at 37 degrees are associated with organic material of human or animal origin. So these are usually pathogenic. Usually, ito na yung mga feces coming from them. While for the bacteria growing at 22 degrees, these are your saprobic or your normally bacteria that inhabit water. So they are derived from soil and vegetables. Unusual decomposition lang. For the presumptive coliform technique, just like what we used, your medium, we use your lactose broth, and then alternative medium for it is your double strength McConkey broth. So your result is interpreted as MPNs, or your most probable number per 100 ml. So in our experiment, we use lactose broth. Positive result would be effervescence in the Durham tube, presence of gas, and the change in color of the medium. So it goes to proof na it's acidic. For our next test, we have your Ekman's test, or the differential coliform test. So this looks like your completed test. Parang ganun lang siya. So it is used to determine whether the coliform bacilli detected in presumptive test is definitely E. coli. So tubes exhibiting gas in the Durham tube are deemed to contain E. coli. Again, positive result sa Durham tube is yung bubbles. And then the presence of E. coli can be further confirmed by testing for your IMVIC. Again, what's the result? It would be positive, positive, negative, negative, positive metal red, then positive yung indole, and the negative yung Hooks-Prescower and your citrate. You can also check for your grab stain if it looks like your E. coli. Next, we have your detection of Clostridium perfringens. Again, what other bacteria can be seen is your Clostridium perfringens in your water. So we have a test for this. We have a specific test. So only if your Clostridium perfringens is detected in the water sample, it suggests that fecal contamination is not recent. So medyo matagal na siya. So varying quantities of water sample is incubated in litmus milk medium. So this is for anaerobes at 37 degrees for 5 days. The positive result would be a stormy cloud formation, and that would definitely reveal the result, which is the bacteria present is your Clostridium perfringens. In the next slide, I will show you what it looks like. So here's what it looks like. And stormy cloud siya. Okay? It's very different from all of our tubes. Kasi all of the tubes that we uses Durham tube para yung presence ng gas. Doon tayo nag-rely with this one and the wool medium.